Wait.
Yeah. So Fanis, we used to make a record. So, but if you have something like unpublished data that you show and you want to it not to be published in the YouTube after that, then it can cut. We have a film editor that can cut some pieces out. So you just say, or we can just not upload the record if you think it's better. So feel free.
Okay. I hope I behave.
Yeah. Okay.
It's very professional. If you want, we can change your words.
Yeah. Well, okay.
Improve your results.
So it's a pleasure to have Fanis here, and in fact, most of the time I used to make a presentation targeting not the old guys, but the students who are going to see it somehow, and to make a personal account of how I see the person that is there. And Fanis is not only someone that I have collaborated or which career I have followed during the time, but it's a friend. So that I know since 2000 or 90s, I don't remember exactly. I think it was in the 90s still. So more than 20 and something years now. And he was at that time doing his PhD. That was a double PhD between Guelph University in Canada and Max Planck in Germany, with Drosophila and with oxidative stress in Drosophila at that time. And that was the point that made us to enter in contact. That was initially made by Clarissa in a meeting, in a free radical meeting, so something like that. If I'm not changing the... Oh,
Oxygen
okay.
Society meeting in
Yes.
New Orleans.
Okay. And at that time, so he was not only a smart and talented student, but he was someone that called my attention wow. That because he was unusually mature about, he was very critical about society, the world, from the political and from the life point of view, but also could match that thing, being critical about the science, about the system, but also being able to work and to make it compatible with talking with people with very different point of view. So he was also at a certain time tolerant with people, not necessarily with ideas. So he was very prone to discuss things and very clear and strong in his point of view, but at the same time very open to discussion and very engaged in social things at the same time, but still keeping at the same time very strong levels from the scientific point of view. So he was together with very strong advisors, both from Canada and Germany, and in a very good environment. And he was really an outstanding person as a student since that time. And since that time, it was interesting that he was working in the 90s in oxidative stress and could keep that track. But after that, he moved to iron and then to iron metabolism that was still related to oxidative stress, to cell biology, redox process, like any iron biology person, and then moved them to Mexico and then to United States in NIH, working with people that we also know. He was had an appointment in a lab that was most used to work with mammalian iron, that was Tracy Royal at the NIH, but started a collaboration with insects, with Drosophila background and iron metabolism in Drosophila, and for that started also to work with John Law. That's a person that several people here know. And from that, it was interesting that from iron, he jumped to metal biology in his more independent career there, mainly after a couple of years moving to England at the Queen Mary University. And metal biology not necessarily linked to redox process, because he then started to work with several metals. And this is an interesting point, because it looks to me that he moved to an area that was not so crowded as redox biology is, because it's a lot of people work with free radicals, so it's then you have so it's I think it's an interesting thing to look when you start to search for your own independent path and in the long track, in a long time frame, where you can give a contribution in a place that is relevant from the point of view of biology, and where less is known. So that's a strategy. I never discussed it with Fanis if that was how this was made, but from my point of view, I think that the choice was something guided by that idea. Well, there are so many things to be done here, so little people doing that work. And in fact, he during all the time was a person that did interesting work, basic, with fine technical standards, and with thoughtful contributions in every place. And I think this, especially the last works, I think with Zinc are like something that is proving the idea that, well, looking to those strange metals is something that really pays the price. So you have less people, less literature, but probably a large piece of novelty in what you find. So then, thank you, Fanis, for being here. Let's see. I'm sure you will have a nice.
Thank you, Pedro. This is so nice. And I seem to remember that when I was a younger person, I took advantage, you realized already, of printing a few of the photos. This is 2000 when we met in Brazil. The meeting where I met Clarissa was in 1999. And people thought I was very opinionated back then. So to hear from you that I was open and engaged with the other people made me feel nice, although I'm not sure. This is Tomás, by the way, at that time.
Yeah.
and Clarissa said, you should meet Pedro, you should meet Pedro. So the next year I was there. And then we came also in 2005. So here you are in 2005.
Yeah.
And this is Tomás growing. And in 2005, we couldn't miss the carnival, right? The second time. So here is me and my political alliances there. And of course, this photo is a bit blurry. because in 2005, I was still, so I think I was not focusing on Pedro, but on the lady on the back. I don't know if. But
better.
this was the point of the talk where I was going to say cheers, because obviously from getting to know Pedro, I realized that nothing can be done without beer. And then we came also in 2012 with Irma, who is my wife. So that's another visit. And Pedro has been also in the labs he mentioned. So he came to Germany and gave a talk at the Max Planck, and he came to the NIH. But Pedro, you have not been, we have not managed that you come to Mexico. So maybe we should repair that if possible. So now the more classic stuff with the PowerPoint. I need to share my screen. And the title I gave was From Thomas Hunt Morgan and the Genetics of Eye Pigments to Metal Homeostasis. Right, and Thomas Hunt Morgan, so he says in Science, a fly appeared with white eyes. Normally flies have red eyes. He crossed this male fly to the females, noted on the next generation that all flies were with red eyes. Yes. Yeah, all flies had red eyes, and this was not surprising because of Mendel's law, which were known at the time. But on the second generation, the phenotype comes back. The surprise was that it was only in male flies, right? We know now that he received the Nobel Prize because he linked the white gene to the X chromosome that also determines the sex of the fly. He was able to do that based on the work of Nettie Stevens, who was studying the problem of why, when you cross two mice, the progeny has half male and half female. What was the factor that was determining what is male or female? And she discovered in her thesis, she called it the X body, which we later identified as the X chromosome, which she identified in half of the sperm, right? And assumed that it had to be the solution. Now, Thomas Morgan went on very quickly and started isolating phenotypic mutants. Today, we're going to look at some point in the talk of these first three mutants from his collection. And his students did the recombination experiments that linked already from 1914 that sex-linked genes are linked to the X chromosome. And it took them about 20 years to make these first famous maps on the polytane chromosomes of the Drosophila. Now, this is a year 1935 where two other important Drosophila people come into play, George Biddle and Boris Efruci. I like the way they start this paper, because I think you could ask the same thing today. And their argument was in that paper that we have organisms that we understand a lot about their development. At that time, there was the German school was very good in developmental biology. And then the new field of genetics was with flies and some aspergillus. And they said, well, we need to study the genetics of the development if we want to understand how genes work and where they are active and all of that. And unsurprisingly, they decided to study the development of the eye because of the color mutants that existed in the eye. So this is the imaginal disc that is in the larvae and grows. And here is the dissection from the adult. So this part will give the eye in the flies. And what they did experimentally is they dissected this disc and did transplants. So they took the mutant disc and put it into a wild type host. And then they observed what color was developing on this tissue within the wild type host. And this symbol in their paper is that the expected color was found. But this symbol, which came in 2 mutants, is that you took a mutant disc, implanted it, and then the color was normal, although the disc was from a mutant. And so that meant that the genes were not working in the disc, right? That changed this color. If there are, I hope this is clear, it's an important point. Their conclusion was that Vermilion and Cinnabar were working somewhere else, not in the imaginal disc. And they even suggested that probably they're making some soluble factor that then needs to traffic and be picked up by the disc, and that this factor probably is required to make the pigment. So if you cannot make that factor in the mutant fly, you don't make the pigment. But if you transplant to the wild type host, the factor is made somewhere else, and the eye disc can still make the pigment. That was the logic of their paper. And we're going to talk about these 2 genes too. That's why I'm introducing them. Now, then we have the 50s and we have Second World War. And Second World War is also important because of the technological developments that came first for military purposes and then were redirected into science. One example is the electron microscope that we're going to see today. But what happened in the field of Drosophila genetics was that people started mapping the genes into the pathways, the biochemical pathways that make the 2 types of pigments that are present in the eyes. And then this was for 10 years the work of a single individual, Nolte, in South Africa, who took patiently one by one all these mutants and looked at the pigment cells in the electron microscope. Now, I will summarize the work from Nolte with a paper by Jane Shoup. This is now University of Chicago. Jane didn't just repeat what Nolte did. She added that she looked in the development. So she took, Nolte was working always with the adult flies and looking at the different mutants. Jane went and looked how these granules develop in various mutants. And this is her conclusion. She said there are 2 types of granules that I can see in the pigment cells. One that clearly grows from the ER, and the other one, and she put this question mark here, and this question mark is still there. We don't understand this process, I would say, to this point, because she says suddenly during the development, I see these large structures, and a few hours before, I do not see anything that is reminiscent of them, so I don't know where they come from. These structures have these filaments initially, and then they fill up with pigment. Okay, and for simplicity, I'm suggesting that the 2 types of granules have the 2 different pigments. Now, 1972, insects have pigments also in other parts of their body, not only in the eye. And it was known from the 30s that yellow pigment in internal organs, this is the testis, they also have yellow pigment in the Malpighian tubules, was correlated with reduced drosopterin in the eye. And obviously, if you know this, the first assumption is that perhaps an intermediary in this pathway, which is yellow, is the yellow pigment. But this turned out to be a wrong hypothesis. And what Nikla suggested was that this yellow was because of riboflavin. And riboflavin also remains to be connected somehow in this story. It is not clear how it connects to this time. 1975, and the Sullivans, David and Marie, they propose, by that time, many mutants have been mapped, but there is still several mutants that they don't know why the pigments are affected. And these researchers suggest, well, pigments also have to move from one place to another. For example, remember that soluble factor I mentioned in the beginning that needs to be captured by the eye disc. So you need transporters, you need to move things into granules. And these were the first colleagues that suggested we probably have genes that are important for the movement of chemicals that are important for the formation of the pigments. 1975 is also very important for a different reason. It is when I was born in Canada. And I grew up in Greece, and I studied biology here in Patras. Then, as Pedro said, I did my PhD in the Max Planck, but in collaboration with the University of Guelph in Canada, where I studied oxidative stress. And then I moved for the postdoc in Tracy Rolt's lab at the NIH. Now, I'd like to say a few words. I introduced a little bit of the history of the pigments, now about iron metabolism. So there are a few papers from Donald Poulson in Yale, who was the first person, together with Bowen, studying metals in Drosophila. And 3 or 4 interesting dense papers with first observations, but then they dropped the subject. I'm not sure why or what happened. And then Helen Nicole and Michael Locke pick it up in the 80s because of the electron microscope and because ferritin is visible in the electron microscope. So some of the people who were studying the pigments in mutants, there is a line in their paper, oh, and we can see some ferritin in this granule, right? Because it's electron dense, the iron core, and you can see it in the electron microscopy. But Helen and Michael teamed up and did a thorough study about the ferritins in Drosophila. And then John Law was already mentioned by Pedro. And John Law took a sabbatical year in 1995, I think, or 6, and visited Fotis Kafatos at the EMBL in order to learn molecular biology. So Pedro, you didn't mention nothing about molecular biology, but there is a link there between me and you and John Law and Fotis Kafatos. But John Law went to Fotis Kafatos to learn molecular biology. There is some tension here because the group in Uppsala that started working rather simultaneously on the iron genes in Drosophila, there is a little bit how much they learned from a visit they did at the MBL around that time as well. But this is from the paper of Fotis and John Law that came out of the sabbatical and is the cloning of the ferritin gene. And he says the fact that we have cloned it should facilitate the analysis of insect ferritin physiology. And I would argue that that's what I started doing when I went to Tracy's group. They said in the same paper, it should be possible to identify produced mutants that lack the gene and study the consequence. And that was the major part of my postdoc, was to do these things that they had already envisaged John Law and Fotis Kafatos before. And then from 2013, my good friend and colleague Bing Zhou came to the field. And I'm quite happy to say he says, oh, the works you did are correct. And he says it with surprise. He says, oh, it's not very common that I find papers that what they say is correct. So I was very happy with your work. And that was a good compliment, right? When it comes from somebody who is critical. And just maybe as a prelude, now we're in the omics era. So there is a new kid on the block. I will call him like that, although Kirst is 10 years older than me. But he has a much better funded lab and the strong technologies. And I think we will soon, their first paper came out in 2019. And I think we will start hearing much more often from that lab in terms of iron metabolism. And also one of Bing's students is now a PI in China. And so her name is Gurian Xiao, and she's also starting to produce many papers on metal biology in Drosophila. So that's where the field, I think, stands at this moment. After my postdoc, I did a small stay in Mexico City. That's where I met Irma, my wife, but then I started in London. The opinions that Pedro was referring to early on didn't work out very well in that city. And so from 2012, I'm living more peacefully in Mexico City at the Cinvestav. And with respect to why I chose to move from iron to other metals, in reality, it was that after 5 years of reading, we had weekly seminars on everything that was new on iron in Tracy's lab, right? So after 5 years of reading almost everything that was getting published on iron, there comes a point where you say, I'm tired of iron, right? I want to see something new. And so actually the decision was much simpler, Pedro. I just said, well, let's learn something new. And I think that continues to be my motivation, right? I dislike anything that I consider known, where I have to do work to show, oh, it is as I think it is, right? So that, which is a very important part of science, right? And often full of surprises, right? We are sure something is X, and it turns out not to be true. What we were sure would be X. But until I figure out that X is wrong, I am bored to go and show that X is as I think it is, right? I'm normally the type of person who is looking for a project where something is not known. And to move on. It took surprisingly long that we understand a little bit. I mean, I will tell you what the model is. I will not say this is the definitive what you're hearing now, but we understand molecularly that white encodes for an ABC type transporter. It appears to dimerize either with scarlet or with brown in Drosophila, and the proposed substrates are written here that are required for transport within the two types of granules I was describing before. So that's one proposal of what this ABCG2 type transporter may be active. And obviously the explanation in this model of the phenotype is if you cannot transport the precursors, you cannot make the pigments, you do not have a color in the eyes. Now, when I was at the NIH, Juan Bonifacino was the director of the branch, right, where I worked. And as Pedro said, I went to a lab that worked with mice in a branch that Juan was the director, and he was working with human cell lines. But he had one postdoc before me these are the years I was a PhD student. These are the years where Juan and Esteban worked in describing this complex, the adaptin protein 3 complex, and they did so using Drosophila flies. Now, before I show you the fly work, let me just say this important bit about the AP proteins in case not everybody knows about them, but you have heard about the phosphoinositides and the different type of phosphoinositides in membranes, and the different AP complexes have affinity for different types of phosphoinositides. So that starts making the membrane domains different. And then these complexes also attract proteins that are RAB specific. And through these interactions and the cargo, you have, they are an important part of the protein trafficking of a fly system. So AP1 of any cell, actually, eukaryotic cell. So AP1 and AP2 are very classic adapters. AP3 is a little more niche because it makes these lysosome-related organelles, of which the subject of this talk is about the lysosome-related organelles. And Juan was telling me, oh, isn't the fly model fantastic? I mean, these classic mutants, they are all part of the same complex, right? These are the mutants that the Sullivans had proposed are going to be trafficking mutants. And Juan was very happy that he had molecularly cloned them and described the AP3 complex. And me, the young Fanis, was listening to all this and was thinking, oh dear, I came to the NIH and these people work with problems that are from the ancient history. Like, I am doing important things. Like, for example, I'm using GFP to tag the ferritin. I feed the fly with iron, and boom, you get a specific response in the anterior. And I need to answer difficult question, right? How did these cells realize that iron was there? How was this response evoked? And how do you make this complex, right? And this guy here is talking to me about what Morgan students were doing, and that he managed to figure out what the gene was, and I was making fun of him. And now, obviously, I have changed my mind because I'm giving the seminars linking to that work. Now, these are questions that we haven't answered, even though they were placed around there, around 2006, 2007. I had formulated the questions, and I'm not going to talk today about Karen, but she's trying to use some of the newer technologies to address these problems. But they are important for the talk because we are now in 2008. I have moved to Queen Mary. I have said before that, oh, I want to study other metals, but obviously at the first conference that was local was the iron conference, and there I met with Lucia Gutierrez from Spain, who was a physicist, and they had a method to measure ferritin based on magnetic susceptibility measurements. So I was working with ferritin, and we teamed up. And we did it here. I'm showing a very simple experiment. This is ferric ammonium citrate. This is bath of phenanthroline sulfate and iron chelator. And these are elemental analysis of whole flies. And obviously, if you give fly an iron, you will have more. And if you give a chelator, you will have less. And the other metals don't vary a lot. Now, this was the first time we did, I used this technology to do metal determinations. And the surprise came on a different angle. It came here on this result that I'm showing here. So the next line has a ferritin heterozygote mutant. These values are simply a bit smaller than these. And here we don't see much difference. But this was a third mutant that was analyzed after some advice from Helen Nicole to look at these flies. And the surprise didn't come in iron, although there may be something in iron. It came here. I saw this and I said, well, this is my interest now. I want to understand this difference. It's also one of those experiments where you, although by now it has been repeated hundreds of times, it's one experiment you don't really need to do replicates, right? The 3 replicates here, and the result is dead clean, right? So I wanted to understand. Now, we went on to suggest that this was genetics. I will show you it was in a second, but it was interesting how a single genetic mutation can change so dramatically the zinc content in a fly. And I put this slide knowing that I'm talking to non-Drosophila people. So we didn't only work with Drosophila. We said, well, if a single gene can cause such a difference, what happens in evolution? And so what Mariam started doing is taking the different Drosophila species and asking that question. And I put here what the answer we gave with Mariam. So we are saying, we think that the general trend of what we found was that they are surprisingly similar, those metalomes. And the explanation we give is that an insect, in order to work, it needs certain metalloproteins, and those cohorts are in general similar. And whenever we saw a difference, it mostly has to do with storage, right? Some insects may choose to store a metal, and some insects may choose, oh, I don't need to store a lot. Now, but from this work and from Lucia's experiment that I'm showing here, we were perplexed because initially we thought this was the normal situation because all our fly stocks were like this. But then the wild type flies were like this fumble heterozygote mutant here, okay? So it took a couple of years and some crosses and some measurements for Negar and Bilge to conclude that there is an X-linked mutation that causes a 3-fold reduction in zinc in Drosophila lab strains. I won't bore you with
what is this fumble mutation looks like?
It's a red herring for this talk because it has nothing to do with the zinc phenotype. This strain had a lot of zinc because the X chromosome it had was a wild type X chromosome. All of these strains have a mutant X chromosome. Okay, fumble is on the 3rd chromosome. So fumble was irrelevant to the phenotype. Okay, I have slides for that too, but maybe not the time today for fumble. Being Zhou that I mentioned has a fumble. But for the next slide, I guess what I want to say is when I went to Mexico, I went with this problem. I knew that there was an X-linked gene, but I didn't know which gene it was. And so I started doing what the students of Morgan were doing, the old-fashioned crossings with markers to try to map the gene, and happily it worked. And then the surprise was that it was the white gene. Okay, so the gene that Morgan, the first mutant ever cloned, had well, no color in its Malpighian tubules, but also no zinc stored in these organs. I'll go relatively quickly with some of the evidences. I mean, this is the classic test for a Drosophilist, that if you have a mutant, here is the white mutant, in this case with two transgenes, that when they meet each other, they express the white gene. And so this is the classic rescue experiment, that the zinc goes back to normal levels if you make white in the fly. The parents of this cross, they are low on zinc because they don't have the white gene. Here the white gene is returned, and then you get the rescue. Here is the evidence that the zinc in the wild type animal is in the Malpighian tubules. This is, I think, females, and this is males. And you can see the zinc is in the Malpighian tubules of these organisms. And then some imaging. This is a probe that's commonly used, that when it binds to zinc, it fluorescence. And the lower panel is the wild type fluorescence, and you can see the stored zinc in the Malpighian tubules. This is the nuclei. This is the white mutant, and these you see the absence of these granules. And then this is a GFP attached to the zinc transporter. And interestingly, in the white mutant, it's only on the plasma membrane. And in the wild type fly, it's associated with these granules. And then I said I would come back to these. So I've already spoke a lot about the white eye, the pink and the orange eye. The orange eye was one of the four I already mentioned when I mentioned Juan Bonifacino. These are the four mutants that Juan Bonifacino cloned. They are all defective in zinc. And this is the pink. These are other mutants that are related. So pink is for the block 2 complex, but then the RAB32, the HOPS complex, are involved in making the zinc storage granules. So if you have these mutants, you don't make the granules, you don't have zinc in the flies.
So, just one question. The first, the zinc granules are only found in the Malpighian tubules, or they are found also in other tissues?
In the Malpighian tubules, you have concentrated zinc granules. In other tissues, if you have, you're going to have one granule in a cell, etc. Right? So, okay. So the storage site for the insect is in the primary cells of the Malpighian, for the Drosophila, right? We haven't yet looked in all the other insects.
Yeah. Okay.
The other question, when I discussed white, it had two partners, was to see which of the partners was involved, right? So we just looked in the scarlet and in the brown mutants. And again, the answer was quite clear that the scarlet, so this pathway was the important pathway for the zinc. And this had a very mild phenotype, so it's not exactly the same as wild type here. But that's again, I think I end the talk with a comment there about this pathway, but that's future work. For the simple way of describing this is that if this was the brown homochrome granule in the Malpighian tubule, it has zinc. And then this was what we proposed with Carlos. We are revisiting this proposal, but because of the NICLA paper and the riboflavin being suggested to be the yellow color of the tubules, we said, okay, then the other granule, the brown-like granule, probably accumulates riboflavin, but take that with a caution. And this is just to show, I said we haven't looked at other insects. I was wrong. Well, we haven't, but this group in Australia has shown that zinc is also stored in the Malpighian tubules of the termite. So you can see the Malpighian tubules here in an experiment. And you can see here, I love this photo because it's subcellular resolution and it's looking directly at metals. And at least in this termite, the same cell separates the calcium granules from the zinc granules. Okay, now in the Drosophila, these, I would need to go several slides back, but I'm going to do it. In the Drosophila separates also the zinc and the calcium granules, but it does it, I think here, it does it in different cells. So you see these here are the cells that have calcium here. And these are the cells that have zinc in the Drosophila. Okay, and so, but then in this termite, the two elements are in the same cell type, and the cell has managed to separate zinc almost perfectly from calcium in different granules. I don't think anybody understands how this is achieved, but I find it a fascinating.
That scheme where you pointed the components of the membrane of the zinc granule, this is by genetics, or do you have a proteomics analysis or something like that?
This is genetics.
Okay.
Yes. The proteomics of the granules in insects, it has not been performed yet. It's
Mammals, it has.
Yes, but now in mammals, the corresponding structure is has not been studied in the context of zinc. So there are granules that are full of zinc, for example, and famous granules, the pancreatic islet cell, the beta cell, which has the insulin, is full of zinc.
Yeah.
So obviously there we know a few things. However, I'm proposing that we should check if what you're going to hear in a few minutes is also true or not in the pancreas of the mammal. I don't think anybody has done the bioinorganic chemistry of those granules yet. So up to here, what I was telling you is that we started from a difference in zinc between flies, in whole flies, and we figured out that the zinc is stored in the Malpighian tubule within granules. And then the next question is, how do we regulate this zinc? All right. And again, we came to this through genetics. So if these were the mutants and these were the control flies, and this is just in the same, every line here is a different strain. And these are the same strains measured for iron. So you see no changes in iron. And these were the mutants that I was discussing so far. This is the brown mutant that I discussed, but is a little bit intermediate. But then there was a whole bunch of mutants that were intermediate between this phenotype, these clear phenotypes. And amongst these were the ones I discussed early on, Cinnabar and Vermilion. And now I'm going to put Cinnabar and Vermilion into a biochemical pathway. Vermilion is required for generating Kinorinin from tryptophan. Cinnabar is the step that takes Kinorinin and converts it into 3-hydroxykinorinin. And this first part of the pathway takes part in the fat body, and the Cinnabar is active in the Malpighian tubule. The Cinnabar is a gene that's not highly expressed, so you can take that with a grain of salt. I'm making the assumption because, well, it is expressed in the Malpighian tubule at a slightly higher levels than the very low expression that is ubiquitous. And here are the data that Vermilion mutants and Cinnabar mutants affect zinc storage. And now,
Fanis, may
yes,
I ask you a question
of course.
about the Cinnabar mutant? If I remember, the Cinnabar is the one that has a very, very small eye, right?
Mm-mm.
Which one is this one?
Let me.
Because I remember this one that has a small eye, but it's red. It's not white. Which
There are
is the white eye of mosquitoes. The same gene that Vitor worked.
but the Cinnabar.
no, that's Vermilion. No, that's Vermilion.
No,
What is the
that's-
phenotype?
No.
How the eye of the Cinnabar flies look like?
They look brilliant red because they cannot produce homochromes. You see here the homochromes.
Mm-hmm.
They are the
So-
brown pigment. So if you mutate Cinnabar or Vermilion, you do not make any brown color.
Oh, okay.
And-
That's why
and
it's
be-
bright red.
Right. And because
Wow.
The pterygids.
right, you
Okay.
remember now the two pathways here. So this is the color. So
Mhm,
if you
mhm.
have a Cinnabar mutant, you block the process right here. And so you don't make this pigment and you get only these pigments. And
Okay.
there are the mutants, you say there are more than one. I'm forgetting the names now, but there are more than one mutants that cause a very small red eye in flies.
Okay, thank you.
I forget, but I don't remember anyone that starts with a C. Right. So these are, this is now a fly that is wild type, has this ZNT35 GFP, so has the zinc transporter with GFP. This is a DAPI. This is some autofluorescence that the Malpighian tubule has, and this is a close-up. Now, if you look at the Vermilion fly, you don't see those granules and you don't see any autofluorescence. If you take this Vermilion fly and feed it with Kinorinin, the autofluorescence comes back and you see the zinc transporter in these granules. Okay. Now, this is when I was fortunate that Erika joined actually she did this experiment as well. She joined the lab and she was fearless, right? So Niels is a physicist working at synchrotrons and we teamed up and Erika was dissecting Malpighian tubules, I think around 20 she put per sample. And then that sample was taken to the synchrotron where you get a spectra, which at this energy, the only element that reacts to these energies is zinc. And then it absorbs. And then what happens at higher energies depends on what is around the zinc atom, okay, on the chemical environment of the zinc. And you can see these are normalized spectra, and you can see that the shape in the Vermilion mutant and in the wild type is different between these two spectra. Now, first of all, if you feed this fly with Kinorinin, you get back your spectrum, okay? And then if you, what do I have here? I think, right, the only thing I have here is that we were so fortunate that later we did the experiment also in another synchrotron, and you can see how reproducible these spectra are. These spectra are not easy to get, and so it's quite a luxury to have it from different sources and reproduces. Now, I think I have kept it easy for this talk. The other thing you can do at the synchrotron is you can take chemical substances. And because of the genetics, we guessed that either xanthurenic acid or 3-hydroxykinurinin were going to be in those granules. And so we synthesized the zinc complexes with xanthurenic acid and with granules. And you see here the spectra of the xanthurenic acid. It's quite different than the wild type spectra. I should say here that with the oscillations that exist in this spectra that are not perceived when you look at the spectra, but the machine, if the spectrum is of good enough quality, gives you the oscillatory behavior that you see in this graph. From this oscillatory behavior, the theoretical chemists can calculate the distances of the different atoms from the metal, okay? And this is how you combine theoretical calculations with experimental evidence, and you get what the structure is in the absence of a crystal structure. And so this was done with xanthurenic acid and with 3-hydroxykinurinin. You can see that the spectrum with the 3-hydroxykinurinin looks much closer to the wild type spectrum. And in fact, what Niels did was a simple addition of spectra, and he could get a superimposable spectrum where he takes the vermilion background, adds the chemical spectra, and gets the actual spectra of the wild type Malpighian tubule. Concluding at that stage that you have 3-hydroxykinurinin linked to zinc and xanthurenic acid linked to zinc within these granules that I'm showing here.
But what is transported is the complex of zinc plus xanthurenic acid, or
no, because I can say no because of the affinities. We have done quite a bit more work, and the affinity of zinc to the 3-hydroxykinurinin is not high enough to assume that they will be together, except in an acid environment of a granule.
Well, but the complex is with the xanthurenic acid, no?
Now, let's discuss this in a few moments. After I say why this is important, I come back to the problems, and let's discuss it then, because this image I have here is wrong, and I'll explain why it is wrong in a second. It was if you assume that all of this takes place at one place, and we are sure this is not true, right? So you have, but let me come to this in a second, Pedro. Okay, and we talk about it then. Let me just say that the kinurinin pathway had not been linked before with zinc, and that this, well, in the way we did, but the connection existed. This is a review in 1980, and the experiment here is they are feeding rats with radioactive zinc, and here in the percentage you can see how much zinc is retained in the intestine in diets that are low on tryptophan, and then when they add the tryptophan. Okay, and it shows you that in the presence of tryptophan, the intestine is full of the radioactive zinc. In the absence, absorption, they interpreted it as if absorption is less efficient. Okay, now, one thing I didn't tell you from our studies is that it's sufficient to add tryptophan. So here you have, in the black line, you have low tryptophan in the diet. If you simply add, for a given concentration of zinc, tryptophan to the diet, you increase storage of zinc. Okay, and if this dotted line is where a wild type fly fed with adequate tryptophan has zinc and a little bit of zinc, then you can see that in the absence of tryptophan, it doesn't matter how much zinc you add to the diet, you don't get to this level. And so we are saying that given that there is a public health concern of zinc deficiency in humans, and many programs of the World Health Organization funneling money to add the salt, the zinc salt in food, that perhaps the problem is that those children or populations are not feeding enough protein in order to have a proper zinc condition in the body. Okay, so that's one important question our work raises. But then we were also quite surprised with the COVID. When it started, the first thing they did in China was to take patients and throw the proteomics, the metabolomics, and try to see what is different in the sera of the severe cases. And in this study, it was interesting, they also included severe patients from other viruses that had lung problems. And whenever a patient was dying because the lungs were in bad shape, Kinorinin was high. This was soon established by others, and by now it's one of the hallmarks of COVID that tryptophan goes down and Kinorinin goes up in the sera of patients. The other part that became evident where people looked was that in the population of the patients that were not making it after the infection, many of them were zinc deficient. And so this came out in epidemiology from India and from Japan. And here I show you a result from a smaller study we did in the lab in pregnant women. And again, the severe cases had lower zinc content in their sera. So the message is that in COVID-19, we're seeing an increase in Kinorinin and a decrease in zinc. I told you that in the fly, the fat body makes Kinorinin, but then the Malpighian tubule stores it in this form. And I guess the way I'm thinking about it is that maybe Kinorinin is the hormone that signals, let's take the zinc and store it into the granules. Okay, and I'm very curious to see if this prediction is going to be correct in human physiology, right? If Kinorinin is going to have that function in human physiology. Now, there are three more things I want to say. I don't know if I should stop here or see. Okay, the first one is, again, a bit historical. I'm taking a quote from 1986, but this was known already in the 40s. What? That the synthesis or presence of the pteridins seems to be required for the normal development of brown pigment granules. So what were they saying? That the red pathway seemed to be important for the brown pathway, okay, in the homochromes, right? The synthesis or presence of the pteridins, the drossopterins, seems to be required for the normal development of these granules, of the brown granules. Now, this is a problem I want to attack, and I think is very interesting. I mean, one thing that I'm noting, and that should be interesting to the biochemists, is that this pteridin pathway is what leads to the generation of tetrahydrobiopterin. And tetrahydrobiopterin is a cofactor in tryptophan allow me to make it a mistake now, to the enzymes that take the tryptophan towards serotonin, or the tyrosine towards dopamine, or the arginine towards nitric oxide. So all of these are key signaling molecules. So I'm assuming that one or more than one of these is important in the signaling that regulates this pathway. But where, how, and what, I have no clue. Is this important? Again, I'm making a reference to this paper. I think this is a very important paper because of the COVID, and I'm a bit curious if this prediction that I'm going to make now is going to come out true in the next few years. But this group shows or discusses that Kinorinin, this is known, Kinorinin inhibits T cell proliferation. But what they show in this paper is in a manner that can be rescued by tetrahydrobiopterin. So you see Kinorinin is here. Kinorinin is inhibiting the T cells in the immune system. This is what we see in the COVID patients that die, right? They don't have CD4 cells, they don't have CD8 cells. And in this study, they show that BH4 corrects this problem. So for me, I have been saying to a few people that if I was about to die about COVID, I would want somebody to give me some tetrahydrobiopterin, right, as a cure. So far, nobody has taken me serious with this proposal, but I'm hoping that the group that published this paper is doing the experiments. I mean, they haven't come up neither with a hypothesis nor with another nature. And I'm hopeful one day to see that that's true. Now, the other thing I wanted to say is this was the title of my seminar, a similar seminar. Well, back then we had fewer data that I gave in 2016. Tell me the color of your eyes and I will tell you if you gather zinc. And while I was preparing that talk, I came across a reference that blew my mind, and I'm going to show it on the next slide, which was this one, where the authors concluded that blue-eyed people have less zinc in their iris than brown-eyed people. So I said, this cannot be, etc. So it's interesting. I haven't followed up on this, but I find it interesting. And now I couldn't close without showing what the problems are with the current state. Now, I was alluding before that this complex is formed in an acid pH. I mean, we say these things in the paper. We say this is stable in acid pH and has high micromolar affinity to zinc. So it's not a very strong affinity. This is why I was answering before, since the model is that white and scarlet bring 3-hydroxykinorinin into this granule. We know the granule has the V80PAs. We know it has the zinc transporter. So the idea is that you make an environment that favors this complex. And that's the lysosome, actually you even need the chloride. And so there is, that's a mistake. I tested if it was this transporter that became famous because it senses, it's claimed to sense zinc in the intestine, but those mutants are okay with zinc storage. Now, the xanthurenic zinc complex is stable at neutral pH, is much more, binds to zinc much stronger, 100-fold stronger than this complex, and is autofluorescent. So maybe what we're seeing here is these types of granules that have the xanthurenic acid. And one idea we are entertaining, we are wondering whether the xanthurenic acid can mobilize the zinc that is first stored as 3-hydroxykinorinin zinc. Right, so we think more that the picture is like that. And actually, I bring you back to an image I showed you. And so I would say these granules here are these ones, and these that are more here, this one, that one, are these ones. Okay, and we're working to show that this is the case. And I think I brought this to say the thanks, because I didn't mention this type of work would not be possible without chemists here, Liliana, Alberto, a theoretician, Fabio did the NMR. Liliana is really my Pedro in Sinvestav, right? My friend in Sinvestav, who is broadly interested in many different aspects and very eager to work on strange problems, right? Like the one I was discussing in fruit flies. And then Niels is a physicist, and who was our connection to the synchrotrons. And Erica was the brave person doing many dissections. When she left the lab, I started doing them. And then, of course, my appreciation for her doubled when I had to do the dissections myself. And Carlos did his PhD on this project, and Karen works on the iron side of the lab. And Joanna graduated with a very nice story on manganese. I didn't speak about manganese at all, and I don't think, if Pedro, your advice is go for virgin fields, manganese is a virgin field. The difficulty with manganese is that it is the metal that is left last when it comes to binding affinities. You see, so iron wins it, and then copper and zinc tend to win. And so how do you make tools that are specific for manganese? At this moment, they don't exist. And the proteins that bind manganese are relatively mysterious in the question, how do they get their manganese? Right? What trick do they employ so that they can attract specifically manganese instead of, for example, iron or other metal ions that have similar binding properties? But it's an essential element, they say. In Drosophila, we were unable to kill a fly with manganese deficiency. We tried very hard. I guess we need to continue trying, but we were able to cause a deficiency. We were able to see a biological response because of the absence of the metal. So, and that started revealing fascinating biology. So I think that's a virgin field, and one that if I find the next victim, I will pursue with some help.
Yeah,
and I think that's it. No, yes.
Thank you very much. So may I make a first question? So, because I'm interested. So it's impolite because I should invite other people to make the question. But as long as I know, some people talk about zinc as a novel second messenger. So and you, it seems to me, clearly avoided that question on what zinc is doing. So what's the role for that? What do you think it is?
Right. Now, first of all, let me agree that zinc is a second messenger. I can throw a bomb also to this audience. My pet hypothesis is that when zinc comes in the cell, it tells the cell, stop using your iron and your copper and your oxygen and your mitochondria doing the oxidative phosphorylation, right? Which is the one type of metabolism that is often used, and it favors the glycolytic, mitochondria silent, hypoxic type of metabolism. And one way to shift towards that metabolism is by bringing the zinc inside. Okay, so in terms of ideas, I have many, but in terms of doing
Where
experiments,
you have that idea? So out of the blue? No.
right.
It looks interesting, fascinating, but from where it came from?
I don't remember I guess things build gradually, because I was working on your question, which was, what is zinc there for? And then the first answer is you have all the transcription factors that bind zinc, right? So maybe let me make a step back. To answer well those questions, you need to go back in evolution, right? You need to go when life starts, and then there is very low level of zinc. And then you have a few enzymes that have huge affinities for zinc. For example, carbonic anhydrase, okay, is a zinc enzyme. It's very important for the interchange of gases, right? The change of CO2 to HCO3 minus to H2CO4. It catalyzes those reactions. And that enzyme is an ancient enzyme and contains zinc. So there are some functions of zinc that you require minuscule amounts of zinc, and those functions will operate because of the very high affinities of those enzymes that are ancient enzymes. And those enzymes are a subset and are a minority of important functions of zinc. The majority of the zinc proteins, because we are talking about 10% of the proteins bind to zinc of the entire cell, right? So this is huge number of zinc binding proteins. The majority have binding affinities that are at a level that is regulatable, right? This zinc signaling is important for them, because if you drop a little bit the zinc, they may lose their zinc atoms. And if you raise the levels a bit, then they bind to zinc and have their function. The most famous of those are the zinc finger transcription factors. Now, to my knowledge, nobody has shown a regulation of zinc finger transcription factors because of fluxes of zinc. Fluxes, because a cell changes the level of its zinc, and then that leads to a change in the transcription factor activity. However, there is a very beautiful demonstration that using genetics. So if you mutate zinc transporters, you block the function of zinc transcription factors, right? So the genetic evidence is there in a Drosophila paper. If you're interested, I can send the reference later.
Yeah.
But that is genetic evidence, right? It's a big, if you block the entry of zinc, yes, you can cause absence of zinc in the transcription factor. Therefore, the muscle cannot differentiate in that specific phenotype that I'm discussing, right? The muscle differentiation requires a zinc transcription factor. So if you block the zinc transport, you cannot make your muscle, etc., etc. So you have proteins. I will quote again Bing Zhou. His fascinating story, recent paper, is tyrosine hydroxylase that makes dopamine. That's an iron enzyme. He claims that enzyme is regulated by zinc. So you bring in zinc, it competes with the iron on the enzyme, and it inhibits it.
By binding to the enzyme?
Right, by direct competition, metal competition on the tyrosine hydroxylase. So I guess, Pedro, I don't know how to attack these problems that are more global in their nature. So I talk about them with my friends. I follow what is there in the literature. I try to find inconsistencies with what I told you, right? I want to see a zinc replete cell doing mitochondrial metabolism happily, right? And then I will know I'm wrong in what I just said before. But so far I haven't found those evidences, and so I like to toy and provoke when I can.
Clarissa has a question.
Yes, indeed, many questions, but I will focus in what I'm mostly doing. You mentioned that the zinc is accumulated in the beta cells, and then I quickly Google PubMed it, and I saw that zinc is important for the insulin storage in the beta cells. So then here's the question: what proteins are in those granules that are important, are being regulated with the zinc for the Drosophila? And then it comes, the other question is, what are the other metabolic features of the white-eyed Drosophila or the other Drosophila? What comes together with these different colors physiologically? Okay,
so first a brief history of zinc and diabetes and insulin. The story starts with a guy called Tim who discovered the staining for zinc. He added a chemical sulfide that would precipitate with the metal and give a black color. And so when you have a staining, you start cutting the tissues and staining and see, where do I see zinc? And the most prominent place he saw it was in the pancreatic islets, right, in the beta cells. And this is in the 50s. So there were people who spent their career from the 50s to the 90s trying to figure out what the role of zinc in diabetes is. And there is a nice review around there in the 90s, long review, many studies, but the conclusion is, whatever the connection is, it doesn't look to be so strong. Now, what led the person to conclude this was also the discovery of the ZNT3 knockout mouse, no, ZNT8 knockout mice, which puts, in my opinion, a bomb in the dogma in the field that zinc is important for insulin to storage in the granule, because those animals are not diabetic to start with. They have insulin, etc. Now, when you stress them, they start to have some mild phenotypes, right? Now, this is 90s. Then comes the first global human study where they looked for alleles that make humans prone to get diabetes. And besides, I think the first hit was the ApoE, which was, and the second hit was ZNT8. So this means I was active, right? And I saw Guy Ratter at Imperial College jumping on that boat, and me thinking on the background, this is now another generation of 30 years that are going to conclude with more modern techniques later that the connection is mild. I'm not saying it's not there. And the third thing to say about this is that I am surprised that the insulin glucagon signaling in the insect, I didn't speak about that work, has been implicated in the sensing of a zinc deficiency. Now, I made another big claim, but that's a published paper. What the authors did there was to study how flies taste zinc. And then they realized, oh, if they are zinc sufficient, they don't care. But if they are zinc deficient, then they are able to detect zinc in the food and have preference for that zinc. And then they realized, so there is a signal that tells the neuron, I need zinc. And that signal was the glucagon. In insects, the AKH, right? The adipokinetic hormone, right? Now, I am therefore thinking, now I told you how I'm thinking about zinc and iron. I think zinc says to the cell, do glycolysis, non-aerobic metabolism, and iron tells the cell, and iron and copper tell the cell, give me oxygen and I'll give you ATP and mitochondria. And I'm thinking the glucose, the insulin and the glucagon are regulating the substrate. So I think those two systems, well, which are the two systems? One is the well-established, the insulin glucagon, and the other one is the one I'm considering, which should have Kinorinin as the signal, bring in the zinc, and perhaps either serotonin or dopamine or nitric oxide or one BH4 dependent metabolite telling the system, I don't want zinc, bring in the iron and the copper and so on. And I think the two systems are working together in physiology. But for the latter system, there is no evidence. It's just in my brain, right? So the former, there are well-established physiologic systems. And I'm wondering how much redundancy there is between the two. That may explain a little bit why you don't see the phenotypes when you knock out only one and not the other.
And about this, in the granules, do you think the zinc also in the insect, if the zinc is helping any other protein to stabilize in the granules? Yeah.
Right. Like it does for insulin, you mean? So what I'm doing practically, Larissa, is I'm trying to get access of the pancreatic islands to the synchrotron to check, do I have Kinorinin in those islets or not? Maybe even the answer is known. It is not known to me. But those granules are full of zinc, and the model by which they bind to insulin is 1 zinc for 6 or 2 zincs for 6 insulins. That is very little zinc. You see what I say? But when you do the staining, you have tons of zinc. You have millimolar zinc in there. So my first question is that zinc bound to 3-hydroxykinorinin in that granule? Because if the answer is yes, then I'm on the game. Let's say I will have more people paying attention, right? But if the answer is no, then maybe I'm working on some insecty thing. Then I will have your attention, I hope.
Yeah.
Still.
Yeah. So, but what about the calcium? Because you show that beautiful picture of vesicles that take up calcium together with vesicles that take up zinc, exactly the same shape. So meaning the suggestion, at least, that they are close to each other, but different regarding the type of ion. And zinc and calcium are very close to. No?
No. The last statement I disagree. Up to the last statement I agree, but the calcium is much bigger, much softer. I wouldn't call it similar. It doesn't have d electrons. That I mean, the zinc is attracted to sulfur. The calcium wouldn't care. So they're not very similar. Calcium and zinc are not very similar. Okay, now you cause me trouble. I'll tell you why you cause me trouble. One reason I'm not studying deeply the calcium granule is because Drosophila has made me the favor to put it in a different cell than the one I'm looking at normally, right? So then I delegate the problem to the people who study stones. There are 3 or 4 groups that use the Drosophila Malpighian tubule to study calcification. I am not saying that zinc is not involved because, interestingly, even in the calcium granule, the core is a zinc core. So this is, again, an interesting and not understood phenomenon where the seed to make the calcium granule is a zinc seed. Okay, and the other information I have from the zinc meetings is also for the people who do oxygen hypoxia regeneration, and therefore are interested in the mitochondria that go wild in this when the oxygen comes back. And part of the apoptotic mechanism is linked to calcium. But before that calcium wave, there is a small zinc wave, the zinc signaling that you were referring to. And people who have inhibited that wave see also an inhibition in the calcium wave. So I am convinced, and by the way, the genetic screen that looked for mutants that would stop the making of the stones brought around the zinc transporters, which was confusing again, right? Why do you have, but these are published data. So if you cannot put zinc into the granules, you appear not to make these big calcium stones.
Mm-hmm.
Why, I don't know yet.
So,
I haven't gone deep in it, but I saw it also. There is a guy studying this process in macular degeneration. So we have an epithelium behind the retina that has all these things you study, Pedro, the lipofusins and the accumulate with age, and then they cause damage in that epithelium, oxidative damage and all of that. Now, the medium stage is calcium dependent, but the early stage again is zinc dependent of the disease. So the first metal that accumulates there is a zinc. And the zinc autofluorescence. So I am again there. I am thinking maybe it's the same compounds we are seeing in the Malpighian tubules that start the process.
mm-hmm.
But yeah,
You didn't answer the other. If the different colored Drosophila, if they have differences,
right. So for example,
or metabolic.
yes, the white mutant, for example, has 40% lower dopamine, serotonin, all the primary amines.
Mm-hmm.
What else?
They eat more, they eat less, they are more active. You know, I want to know about if they are fat or not.
The vermilion mutant eats less.
Mm-hmm.
And I mean, that was a point that the reviewer was fighting us until the last moment. And those experiments were done in response to review. So they are not, let's say, our deepest experiments. We haven't studied, we haven't repeated that experiment enough. But the preliminary finding was that we could not rescue the feeding defect of the vermilion mutant with Kinorinin. So I showed you how we can rescue many things with Kinorinin, right? The vermilion mutant is, you cannot make Kinorinin. So instead of rescuing it with the classic genetic way, we were rescuing the mutant by giving Kinorinin to the food, and that would rescue a number of phenotypes. When we did the feeding assay, we saw that the vermilion eats less, and then we add the Kinorinin, and it didn't start eating more. Of course, you can start asking about details of the experiment, and that's why I say I'm not ready to hold this this ground for too long. But if you care about do they eat a lot, they don't. And then Clarissa, I'm embarrassed, but we did the Nile red stain that to look at the lipids on the white plus and the white minus. I mean, I say we, and Carlos did that, and he saw a difference, but I don't remember the direction. So this is very, very, and he didn't have he had forgotten to bring the photos, so he just told us, and so I didn't see the result, and I don't remember now what he said. But yeah. I mean, I'm sure that there will be. I mean, when I'm saying the metabolism changes, then it's obvious that the lipids will also follow suit. I mean,
Yeah. Okay. Do you have any guess about why in Malpighian tubules?
Yes, because this, I'm thinking that the way the insect deals with the zinc is it takes it in the intestine, in the hemolymph it moves around, and then it goes out through the Malpighian tubule. So right, and the question I don't have an answer to is, once you have it in the zinc granule, which for me is the 3-hydroxykinorinin granule, can you remove that zinc and bring it towards the body? Because it is clear that you can throw it out. And my favorite.
Clear. You find a lot of zinc in the urine.
The reason it is clear is the transporter is there and it's inducible also. When you have excess zinc, the transporter is inducible on the apical side of the cell. So, I mean, I don't know of anybody who measured the zinc in the feces or whatever, but the response is there in terms genetically, right? The exporter is put at the right place when you have excess zinc. So that makes people think, okay, that zinc is going out. Now,
but maybe going out back to the hemolymph.
Well, it's polarized. It's polarized. Right. It's in the lumen. But what can happen is that further down, it happens with calcium. That's why I think it can happen with zinc. Further down, so in the part of the Malpighian tubule that's near the intestine and in the hindgut, you can have reabsorption. Right? In the case of calcium, this has been shown. In the case of zinc, it has not been shown, but I think it is if you ask me how would I bring back that zinc from the store to the insect, my favorite route would be you will put it in the lumen of the Malpighian tubule, but then you will reabsorb it further down, right, into the hemolymph, either through the hindgut or the rectal pads or the ureter, right? Just before the ureter, the structure of the cells is different than in the rest of the Malpighian tubule.
Okay.
What is the strongest evidence that xanthurenic acid also has a role in iron metabolism?
Not very much, just it binds iron.
Okay, I have asked
zinc as well. So
yeah, I have asked the chemists to figure out in detail the affinities and the, so that, but do any biological argument that xanthurenic, I thought that we had a few biological arguments that it may be important for iron.
We didn't, at the beginning, I just on purpose avoided iron because that was John Law's area. So then I moved, I made everything on iron, on him, just not to get on iron. And at the end, that became a kind of just an habit, a way of thinking. So, but iron is possibly at least as important as him. And I didn't jump into that. So it's something that
now
didn't look at that time.
Now, Kirst that I mentioned, the guy I mentioned that in 2019 has his first paper on iron in Drosophila. He is very interested in this iron versus heme debate, and he has mutants that are rescued by iron and not by heme, and mutants that are rescued by heme and not by iron. So yep. There is a clear, if you wish, distinction between the two. And something for Clarissa too, because she one of your first papers was the heme binding protein in the hemolymph, no?
Yeah.
And one of the provocative ideas, yes, of the tick. But one of the provocative ideas, it has not been published. The person who's trying to publish it probably hasn't succeeded yet to cross the peer review barrier. But the argument is that the classic iron cycle in the human body assumes that in the spleen, all the iron goes to transferrin. You know, it goes first to ferritin and then to transferrin and goes in little bits. And then you have to make again heme in the developing erythrocyte. Mm-hmm. And there are people now out there thinking this is not how it's happening. And a lot of heme may traffic directly and get incorporated into hemoglobin. Right. But this is just debated. I don't know of anybody who has challenged the model, right? Because if we teach the iron cycle, we do not teach heme as part of the iron cycle. I mean, we teach it in the sense that it gets degraded in the macrophage and it gets resynthesized in the developing erythrocyte. And so I said something for Clarissa because the idea, no.
Yeah, but then this matches perfectly with the paper, with the work of Liliani, that was our student, PhD student, from me and Pedro, that in developing erythrocytes, you have inhibition of, no, indeed was inhibition of hemoxygenase.
Yes.
Right, you don't want hemoxygenase there, obviously.
Yeah, but then there's also a link with the enhancer transporter.
FLDCR.
Okay,
yeah.
of the
so
heme.
then it seems that the cell is enhancing, it's not degrading heme and making more heme, but then it also receives heme. So this is a cell that will not degrade heme when the heme enters, but reuse heme.
But do we know by now who traffics the heme in the blood? In the human blood, who is which is the protein?
Presumably.
Mm-hmm.
Yeah.
Okay.
But just before we finish, just one thing. We showed the paper we published in January that the Kinorinin monooxygenase, the Cinnabar mutant in Aedes, it's not a very strong phenotype, but it's a deleterious phenotype. But in Anopheles, It's a strong phenotype. The guy just dies when it eats blood. Interesting, in the Hodgins, it's also a strong phenotype, but after it mounts from the first to the second instant. So I think it's an interesting model, blood-sucking animals, because they have an extra, a very large apport of tryptophan from protein degradation. And this is a kind of critical. So maybe it would be interesting to see why the Anopheles mosquito died and why the blood-sucking guy, the Hodgins, died.
And
I'm going to show you in a couple of days. We're making an interesting experiment with a chemical inhibitor of
the Cinnabar. Right. And keep your eye. I mean, I know you study on that, but the microbiota and that pathway are very commonly invoked.
Yeah.
So when you say I have two mosquitoes and one responds and the other doesn't respond, maybe the other has a bug that helps with the condition. I don't know.
Maybe. But in the Hodgins, the drug, essentially, just to make a spoiler, the drug kills in a couple of hours.
The bug.
Yes.
And this
I guess is in the Malpighian too.
Right. And this is
to
dependent on the Rodnios feeding or not. So it needs to feed in order to die.
We gave the drug along with the blood. It's interesting. So we may talk after. Yeah.
May I ask a question, Pedro?
Yes.
Oh, first of all, Fanis, thanks a lot for a nice talk. Very interesting. Unfortunately, I lost part of your talk, especially when you were talking about supplementing tryptophan. I couldn't see exactly what was the experiment, but supplementing tryptophan, there was a consequence that was increasing Kinorinin, I think. Is that right?
We assume that, but we didn't measure that. Let me show you. The data. So, and make it bigger. How do I zoom? So the first experiment here, this is the wild type fly.
Yeah.
This is the tryptophan feeding, and this is measuring zinc. So giving to a normal fly tryptophan, you increase the content of zinc that it stores.
Yes.
Now, here I'm showing this again. On this axis, I have different concentrations of tryptophan in the diet. And the red and the black lines have different content of zinc in the diet. But the message is more tryptophan, more zinc. And one could debate this point, because when you come to these concentrations of tryptophan, you have to start worrying about how you solubilize the amino acid. There is I read the papers after I had done the experiments of the problems that can occur once you reach these concentrations. And then this is the other evidence here, that here I'm increasing the zinc, but on the black line where you don't see important increase in the zinc, it's because it's low on tryptophan, the media. If the media has plenty of tryptophan, then you have a linear almost relationship between zinc in the diet and zinc in the fly. So I guess the message is that tryptophan will lead to enhanced stores of zinc. That's the first message, or that's the direct interpretation. Now, I took it a step further in my talk because I suggested that tryptophan will cause, I mean, there is the vermilion, which is in humans, it would be either indoleamine oxidase or tryptophan dioxygenase. So there are three enzymes, actually, 2-idose and 1-TDO in humans, and their role is to convert tryptophan to kinorinin. And my suggestion is that kinorinin gives a signal to move the zinc from the circulation into the store, into the granules. That's my suggestion. And so one of the reasons why you have more zinc when you feed with kinorinin is because you store more zinc in the granules, because you make more of that signal that says, please store your zinc in the granule. That's the way I'm thinking about it, but.
Part of my question was involved for what you said. If you have considered the possibility that the kinorinin being transported by exosomes to have their effects, because I'm not sure, but I remember reading a paper that involved the exosome as the vesicles carrying kinorinin. I'm not sure. I have to check that, but that's why I'm asking you if you have considered the involvement of exosome in the metabolism of. I don't know.
I like the question. The first thing I did while you were asking it was to put exosome and kinorinin in PubMed. Do you recognize one of these three papers?
Mm-hmm. No, I don't remember really.
Because my answer is I do not know. I do not know if kinorinin is released with an exosome or on its own.
Mm-hmm.
And but we know that you need kinorinin in order to initiate the formation of these lysosome-related organelles. Okay, so and this is a puzzle. Why do you need kinorinin to make these lysosome-related organelles? We do not know the answer to that question. And I usually add, we do not know what the transporter is for kinorinin or the receptor, but you are adding a possibility that the kinorinin comes already with its vesicle.
Yeah.
And this is a very interesting suggestion, because we know that the ATG8 part of the autophagic pathway is required for the formation of that lysosome-related organelle. So
who knows?
Yes, so you have a what I would call a receptor membrane.
Yeah.
And therefore, if a vesicle came with the kinorinin,
exactly, yeah,
it makes sense. And so if you fall on that reference that says that kinorinin is in exosomes, by all means, I'd be interested. Tell Pedro that he sends it to me or something, or you. if you fall on the reference again, or if you remember who said
Yeah,
it.
okay. If I find, I will send to you. Okay. It was a couple of years ago, but so I don't remember. Very nice. Thank you.
I have a question. Do you remember when we met each other in 2000?
Pardon?
I would be surprised, but when I came as a boy to the department of Pedro, he introduced me to you, but I don't think you would remember me since then.
When it was 2000?
2000.
22 years ago.
Maybe, yeah. Well, probably you were younger, so.
Okay.
Oh,
Thank you for the
just
question.
Fanis, I can email you the data, but there's a paper showing and that COVID exportation has to do with this that Masuda mentioned, that COVID depends on the lysosomes to be treated. And we have a project now looking at the caveolin's role in the COVID.
But wait, because you work with humans, right? Because I know of someone in Sinvestav who took the antibodies for caveolin and applied them to insect tissues, and they work fantastically. And so he keeps talking about insect caveolin.
Yeah, so then, there's no caveolin in insect.
Right. Huh? But then he says there must be, because I took the antibody and I see phenomenology that's consistent with caveolin, right? Which is quite an interesting situation to be in. But yes.
Yeah, I have a student now working in this caveolin and COVID in mammalians, for sure. But then I looked at some point and there's no caveolin in insects. This is very weird. There should be, because it's a very basic protein.
Yeah.
Right. So I think there is, and we just don't know the identity.
Mm-hmm.
Yeah. So I think it's about midnight for you, no?
For me, it's 11.
Yeah.
I thought you went back to Brazil, Clarissa. It's
not yet.
I'm
trying to have a bit more time here because of COVID.
The rumors go faster than you. Then I'm going to go back to my source and say, well, I was surprised.
Okay. So I think we can.
Thank you so much, everyone, for being here so long.
Yeah.
Thank you, Fanis. Very good.
Bye.
Okay.
Bye-bye. Hi,
Bye.
people.
Bye.
Thanks a lot for the
Thank you.
presentation.
Thanks a lot.